All right, welcome. So I'll talk about deciphering data, statistical analysis with MAXQDA. I think this is a really interesting topic because it sort of opens the doors to maybe a new way of thinking about research or lines of inquiry or ways to conduct analysis that maybe you haven't thought of before. Um, I'm at the University of Michigan and I'm a methodologist specialized in mixed methods research. So most of what I do is combining quantitative and qualitative methods. And what I'm going to do is alternate periodically between this slide set and some demonstrations in order to show you some of the statistical analysis tools and the ways you can incorporate variables within MaxQDA. So bear with me as I'm going back and forth a few times. I think it'll hopefully make it a little bit more realistic when we move over to MaxQDA for some of the demonstrations. Um, so some of the major features we will cover are the use of variables and then moving on to the stats features. And the reason I'm starting with use of variables is because you can launch your statistical analysis from variables that are in MaxQDA and attach to things like documents, say transcripts, for instance. So we'll start with that. We'll then move into some stats specific features, starting with basic descriptive statistics, and then moving on to some inferential tests that you might be able to conduct as well. So things like correlations or t-tests, analysis of variance, and so on. And we will look at some of those. So first of all is working with document variables within MaxQDA. So I want to make this distinction really clear. You don't have to have variables attached to your data sources in order to use MaxQDA stats. You can use an external file, and that's certainly okay. For the work that I do, more mixed methods work, I typically do have variables that are attached to documents. So things like interview transcripts, characterizing people, or maybe it's scores or something like that, that we have also collected. So what I will show you first is how to add document variables. You can do this manually. So we'll look at creating a variable, specifying the type, that sort of thing. You can also have data that's stored elsewhere and you can import that. And there's a really helpful wizard that will do it. So the idea is that you can have data stored in another spreadsheet and it can look at a unique identifier on the spreadsheet and then attach and import those variables to the same unique identifier that represents a document in your document sources in MaxQDA. As you're adding variables, if you do this manually, you can establish types of variables, which I'll show you in a minute. This whole idea, though, of bringing variables into MaxQDA, and one reason I like it so much is because it unlocks additional analyses that you can conduct and additional features. So, for example, you could activate your documents then by a variable. You could say that I want to activate everyone who identifies as male or female, or I want to activate individuals who are less than 35 years old, or I want to activate in more of a mixed method study, those who scored highest on a performance measure or something like that. So we'll look at those features as well. Another thing that you can do then when you have variables within MaxQDA is that you can display variables in retrieve segments. So this is really facilitating a true mixed analysis. And I know we're focusing more on stats and we'll getting to this, but I find that having when I'm using a MaxQDA for mixed methods analysis, having those variables visible and those values visible can be really, really helpful. So here's an example from one of my studies. I'm going to show you examples from real studies, real data sets that are identified, research that's been conducted. We'll look at this in just a moment. But on the upper left, of course, are my document sources. So I've activated documents from, in this case, it's an intervention group, and I've activated some codes. And if you look down at my retrieve segments window, you will see here, it's where the arrow is pointing, that I have variables attached with that particular document displaying. So I can see for this first segment of text, That this is from a 26-year-old. The gender code, it's 0. I coded 0 and 1 for male, female. And then I have a score and an assessment that was conducted. And then this last one where it says equals 2, that's a categorical number of someone's score on that assessment. And we'll look at this in a little bit more detail when I flip over to MAXQDA. So let me go ahead and do that right now before we actually move into the stats module in more detail. So again, I will do this periodically, going back and forth between the two, just so that we can really delve into these features. Just a little bit of background context of this study. This is a study in which individuals, and in this case, it was learners, individuals who are in training to become physicians, were randomized to one of two groups, an intervention group and a control group, all for the purpose of providing training and improving communication skills. The intervention group was using a virtual human program, very much virtual reality oriented and adapted, so that someone was practicing communication. And in the control group was more of a static learning module, like you might see with videos and annotated PowerPoints and that sort of thing. We had quantitative post-test scores. We'll look at a bunch of these quantitative measures in just a minute, comparing the intervention and the control groups. And that is something that we'll kind of focus on when we're continuing to look at this and look at this analysis. So this study hopefully has a little bit for everyone. It's somewhat health sciences related. It's bringing communication, so social science. It is testing a virtual human program. So there's clearly an information systems and computer science aspect. And then to top it all off, of course, we're using quantitative and qualitative methods to do so. And then there was an educational component too, because we were trying to provide training. So hopefully this appeals to people from many disciplines. These on the upper left, of course, are my documents. So I have it separated into two groups. The empathic group was the training group, and this called SBAR, that was my control group. I'm just going to show you how I've, by activating some of these, and then in retrieve segments, we can see these variables that are now displaying. So this, again, can be really helpful looking at retrieve segments. You can contextualize if of course, that is your goal. But now let's go ahead and look at variables in MAXQDA, what they look like, how you can actually get variables into your file. So I clicked on this first button in the up. I went to variables and clicked on this first button in the upper left, list of document variables. And I'll make this a little bit bigger. All of these that you see in red, these are system generated variables. So they're variables that MAXQDA generates. So for example, coded segments. That corresponds to this number of coded segments that you see attached to each document. So that's something that MAXQDA has created. If I scroll down a little bit, all of these in blue are variables that I have added to this data set. So they're all things that I have created. And I created them by importing these variables. I had it stored elsewhere, and it just looks for this identifier. So each one of these was a row in that Excel sheet. It matches it up with a correct transcript, and it imported it. So if you look through this list, you'll see a lot of different variable names along with the variable type, whether it's going to be displayed or not, that make a little bit more sense later. And then over here, you can designate something as a favorite variable. And because I've designated some of these as a favorite variable, that's why they are appearing down here in retrieve segment. So you can toggle this on or off by clicking on this icon, display favorite variables. So it's off and now it's back on. Again, this is really helpful for starting to characterize your data. But I'll come back to the list of document variables, just show you briefly. In addition to importing it, you can do this by hand. So I can come up with this icon for a new variable. I can type in what my variable name is going to be. So I will call it the COM score. Let's just say it's a post-test measure. You can select your type. We'll say that this is an integer. You can specify if there are missing values as well. So if you've coded some of your data as missing, you can specify what that would be, and you could indicate if it's categorical or not. So I'll click OK, and we see now this COM-score is appearing here at the very bottom. So this is, if anyone's used SPSS, it's kind of like a variable view. Let's switch over and look at the data view. It's called the data editor. So we can do that in a couple of ways. We can come back to this little icon up here at the top and switch over to the data editor, or back on my variables menu in the ribbon, I could click on the data editor. So here is this data editor, and it looks very much like a spreadsheet. You know, it looks like what you would open if you were opening Excel file, or if you were opening SPSS or something like that. It's very much like what you would see there. So this is just an introduction to variables, what they look like. I'll just kind of scroll through so you can see what all of these look like. You know, nothing unusual, but essentially a column is representing a variable. If I should say that if you want to edit this manually or change it, you could. So I could come in here and I could enter in my own values. This was categorical, so I could select a different value instead. So you can edit and also change this manually. Let's now just go back to the slides for a moment and spend a little bit more time talking about stats and the MAXQDA Stats module. For those of you wondering, you know, why you might want to do this, here's a few reasons that I can think of. So for one, for mixed method studies, you know, of course, this is ideal. You could house all of your data in a single program. For one, it could prevent just the need to go back and forth between multiple programs like MAXQDA and SAS or Stata or SPSS, whatever it is you're using, or maybe it's just Excel, by having it in a single program. So that's great. As you're running these stats, you can save your output. You can pick what output you want to save. Say you ran descriptive statistics, you can choose to save that and then easily put it into reports. I think this is incredibly helpful for things like a thesis or dissertation, so you can grab these tables along the way as you're doing it, but really for anything you might be doing, if you're preparing a manuscript, an evaluation report, whatever the case might be. But where I think it's also helpful is for more of a mixed methods analysis. So, and you don't have to be doing a mixed method study to use the stats module or find value in it, but it's one area where I see that value and find that value. So for one, you could be running statistical analysis on the same participants involved in the qualitative component of your study. That can then be really helpful because it can enable additional features, some of the mixed methods features that are not covered in this session. But moreover, the idea of going iteratively back and forth between your qualitative and your quantitative data. So maybe you're conducting your qualitative analysis, you're seeing a pattern, you're maybe starting to wonder about some things, and maybe that's raising an idea of a hypothesis that you would want to test, or way you want to categorize your data descriptively, see if there's a difference, for instance. You could then go right over to the stats file and run that. So kind of this idea of following a thread. So as you have a new question that arises, you could do that. Similarly, you might be conducting your statistical analysis in MAXQDA, and this is something that happens. And that leads to new questions that you might want to explore within your qualitative data. Maybe you want to code something from a different perspective or add a new qualitative code. But you wouldn't be able to do that, I would say, as efficiently if you have your data in another program. So here are some of the features in the stats module that we'll go through. You can use internal variables, and by that I mean variables that are stored within MAXQDA, say they're attached to documents, or you can be using external data as well. So you can use a file that you're just bringing in for the purpose of using it on the stats module. So within descriptives, you can conduct things like descriptive statistics, think means, medians, standard deviations, all of that kind of stuff. You can look at frequencies of categorical data. And again, we'll look at this. You want to know how many individual participants based on gender identity or based on region they lived in or something like that. or maybe you have a categorical or an outcome variable that you're using, as I do in some of my studies. You could also use this for survey items. So for example, if you have multiple response items or a matrix item, so think of a set of related items on a survey that are all comprising a scale. So these sort of go together. You could analyze that as well. Multiple response. So this is, more of a check all that could apply in a survey item as opposed to selecting just a single answer. And then that takes us to inferential test. And there are a lot of options here. Just for full disclosure, this MAXQDA Stats doesn't do incredibly advanced statistical analysis, but it does handle all of the basics, and it does so really quite well. So things like running crosstabs of categorical data, t-tests, or man-Whitney views, depending on what your data types are, running analysis of variance, correlations, different types of correlations, cluster analysis, and then also some scale analysis. It will not do things like structural equation modeling, which often requires an advanced program or an additional add-on anyway of the stats programs. But it really handles these foundational kinds of analyses. So how to use the stats module. We'll look at it in just a moment. I'll open it up, but essentially you see something that's kind of similar to what we've seen already in the variable view or in our variable list, in that you can have your data editor. So that's what's looked like a spreadsheet that we were looking at. You can look at your variable list, so very much like the list of variables we looked at, but then also you can have the output viewer. So when you run an analysis, you can save output. So I want to save my descriptive statistics or my frequencies or a correlation matrix, for instance. You can do all of that within MAXQDA Stats. And again, we'll go over in a moment, and I'll show how to launch that, and we'll go through this one by one. But let me just first introduce descriptives before we go back over to MAXQDA. So you can run descriptive statistics in your data. I asked for basic descriptive statistics from that same project that we were just looking at, and I asked for it for 5 different variables: for age, a verbal score. This was a test score someone had before they went into training, physical science score, biological science score, and then undergraduate science grade point average. So kind of a culmination of grades through multiple courses. The descriptives comes out in this nice table. We see that there is a sample size, the N, the mean, standard deviation. It shows how many are missing and what percent were missing. So it's a clean output, and you can choose to just look at it like this and do nothing else with it. You can also save this output. So I'll show you in a minute, but there are a couple of little icons that are available here. And this first one, it looks like a page with an arrow going into it, next to the page with 2 arrows going into it. Those will basically take this output and they will put it in that output viewer. So you can save it along the way and then decide what you want to do with it. And the output viewer looks like this. So, and we'll again look at this, but this is just something that accumulates. So this one on the very bottom is the descriptives that we just looked at that I saved. So age, verbal score, and so on. One thing that's really nice about this is that you choose what you want to save in the output viewer. So it puts you in control of that. I also use SPSS for statistical analysis. And one annoying feature there is that it just saves all output. It gives you a full record. And then you have to go back through and delete what you don't want. Here, you have to remember to save it, but it gives you full control of what you want to save in the output viewer. I find that incredibly helpful because I'm not perfect. I make mistakes. There are times where I might ask for descriptives on these 4 variables, but I forgot to include one. So rather than having to go through the output and change and delete, figure out what was the right one, I can just include the ones that I actually do want to save. So let me jump over before we go through some of the more inferential tests, and we'll look at MAXQDA here. So to get to the stats module, there's a little option on my menu. It opens up the stats ribbon, and then it gives me options. I can start with all documents. So that means that it's going to take the variables that have been attached to these documents in my document system. I could start with just activated documents. So that's something that's also interesting and potentially helpful. So right now, I just have my intervention group activated, not my control group. So maybe I want to look at them separately. I could choose to do that. I could start with activated documents and codes. So, only those that have had those intersections. Or I can bring in an external file as well. All of those are options. I'll just go ahead and start with all documents just for our purposes here. And we can see that it opens up this window. And just give me a second. I'm just moving my Zoom toolbar out of the way so I can see the top here. So it opens up this nice window. This is really what the MAXQDA Stats looks like, what you'll be interacting with going forward. So it does give you options to bring in a new external file and open that. You can choose in the menu what you want to view. And then the rest of these are getting into more of the analysis that we'll be using. So you can compute or recode variables. This can be really helpful. Let's say you have something that's reverse coded within a scale. Often this is done as part of a scale, kind of as a reading check to make sure people are reading the question and not just answering 4s across the board, where the item is written in the reverse, in the negative. So maybe for the other items, a 5 is positive, but for this one, a 5 is going to be negative, for instance. You can use this to recode those either into new variable or the same variable. So that could be really helpful. But here's our data editor and then the variable list. Again, it looks very much what we've seen before. Just a couple of other differences. You can come here to the measure, and it gives you options based on the Stevens scales of measurement. To select if this is interval data, ordinal data, or nominal data. And that has implications for what statistical analysis you might run. Otherwise, it looks pretty similar to what we've seen so far. Again, it gives me the system generated variables. So you could look at counts of codes, that kind of thing. But then all of these in blue are my variables. So let's first come to descriptive statistics. And I'm going to go down to descriptive statistics and just run that. So it gives me all of my variables on the left-hand side. I can choose which one of these I want to include. So I will include age. I'll include some of these pre-test scores. And then these that start with an at, in addition to this global, those are my primary outcome measure for this study. So that's, and you just either drag over or click the arrow over to bring whatever you want into MAXQDA. There are some options like exclude cases list-wise. So if there's some missing values, it would exclude that, for instance. In terms of the statistics then, it gives you quite a few options in terms of what you want to include. So we have the mean, standard deviation. Note MAXQDA distinguishes between the sample standard deviation and the population standard deviation because it is actually a different formula. Most programs are not that specific. They just give you one of those. But there is a slight difference in the formula to account for degrees of freedom. So that's really nice that it does that for standard deviation and similarly for variance. And then I'll just ask for the minimum and the maximum, and I'll click OK. So here are the descriptive statistics, sort of like what we looked at in the slides. It's a nice table. Excuse me. I can choose to save this to my output viewer. Note, I can select which one of these variables. So I'll just show you. I can choose to save that to the output viewer, and it is bringing it in down here. If you have multiple analyses, you could choose to save them all of the results tables in an output viewer. You could also choose a chart view instead. Now, it's, doesn't make, this isn't bad, I guess. I mean, it doesn't make sense for all types of data, but it gives me this kind of box plot for age. I can come down to verbal score, so on, and look at that. But the charts are pretty nice too. You can, of course, change this, depending on your needs. And then also I'll point out that you can bring this into your QTT, your question, themes, and theories worksheet as well. So it gives you an option to do that. So these are box plots. These are sort of everything plotted, and then it can give me more of a column graph. It doesn't really make sense for this data, but this is what a histogram would look like. So those are descriptive statistics. I'll show you frequencies. So I'm going back to descriptives and then to frequencies. So I will grab some of the categorical variables within the data set, including my categorical outcome variable. I will click OK. And we can see that it gives me just frequency tables. So I can see this for gender, for the module, and then for the score, the categorical outcome variable, high, medium, or low. Again, I can save this if I want into the output viewer. And this is probably a better option where I could, if I click the double arrow in, it will insert all of these results into the output viewer. And it's asking me, do I want to include descriptives as well? I'll say no, because it doesn't really make sense for this. And here you go. All right. So back over to the data set for, or the slides set for a minute. Just a few more tools. For one, you can conduct tests on categorical data. So at a basic level, you could just run crosstab. So looking at patterns between two different categorical variables, but then you can also run a chi-square test to test whether there's a statistically significant difference between observed and expected technically. So I've asked for crosstabs between the degree and then the site. There were 3 different sites as part of this study, and that's what it's giving me right here. So site 1, site 2, site 3, and then I can see how many of each degree type were in each of those sites. I can then also obtain that chi-square. So again, this is more of a measure of associations. So is there a pattern if we look at degree code by site, and it gives me an overall Pearson chi-square. We can see that that was significant. Of course, it's not really 0, it's just a small number that it rounds. And then it gives me some other statistics too, like Kramer's V. You can conduct a t-test also within the stats package. So comparing groups, for instance. So I want to compare my intervention to the control group on the primary outcome variable and see if there is a statistically significant difference or not. And that's what we've done here. This output t-test for independent samples is it's a fairly straightforward output like you might see in any statistical program. A couple of things that are nice, though, particularly nice, is that it does automatically give you your effect size. So that's what this Cohen's D is, or Hedges G, for instance. So you have those as well. Let's say you're comparing 2 or more groups, or you want to control for something else. You can also conduct an analysis of variance within stats. Again, we'll look at this in just a moment. So I've asked for this one of our domains on the post test. It was openness, defensiveness by site, if there was any statistically significant pattern there. So it runs an analysis of variance, gives me my p-value, which is not statistically significant if, let's say, we're using a 0.05. But then it also does, again, give me an effect size, which is the eta square. And then down at the very bottom here, it also will give me several different what are called post hoc tests. So with an analysis of variance, you can be comparing more than two groups, let's say you have three different groups, and of course your first analysis, the analysis of variance, just tells you whether any of the groups are statistically different from one another. And the post hoc tells you which of those are actually different from one another. So it does a very nice job of this, which we'll look at it in just a moment. And then just really quickly, cluster analysis as well before we go over. So here, what I've done is I've dumped in a bunch of different variables and I've asked to look for clusters. Does there appear to be some kind of a pattern or relationship where some of these variables are kind of hanging together, so to speak? So depending on what you're doing, maybe you're working with an instrument or measure, or you have just a bunch of different variables, you need to reduce to fewer variables. This cluster analysis could be helpful. And then finally, you can look at a correlational analysis. So is there a relationship between two or more variables? And then a scale analysis. So it can run things like a coefficient alpha of a scale, looking at how well items are consistent within a scale. This is called internal consistency reliability. And then it also gives me some indicators of which of these items might not be as reliable. But let me just go over to stats for a moment, and time goes so quickly, but let me just show you a few of these. I will come first to this t-test for independent samples. So first of all, I enter in my dependent variable. So I'm going to say it's this global score, but you can have more than one. So, why don't we just do this? This isn't, the best idea because all of these are related to the global score, but you could humor me for a moment. And then I'm going to come to this module, and I'm going to enter that as my group. Whoops, that was not the one I wanted. I'm sorry. Let's do. No, it'll be okay just for demonstration purposes. And I will click OK. And this is it's somewhat contrived because I don't have enough differences within this data set, just the way it's set up. But essentially gives me a t-test saying that there was nothing here, which I would expect. Let me do another example that'll give us results that make a little bit more sense. Whoops, I don't want the document group. So let's do sites, if there are differences between sites. So I'm going to compare site 1 to site 2, and I will click OK. Here we go. This is better. So again, it gives me my p-value, so one tail test, saying what the difference between the means are of the groups. So I can see this, is not statistically significant. And then if I look down here, it's showing me what exactly those means were, and they're very similar. So I'm not surprised it wasn't significant. And that's what I would expect in this case. We would hope that there isn't much of a site difference. Let me just quickly show you the analysis of variance, and then I'll pause for questions. We'll do that exact same analysis that we just ran. But here, I'm going to come back to site, because this will account for all of them, and I will click OK. So here, again, we're seeing that there is some difference between groups if we look overall. And then we have our post hoc test, so we can tell what those differences actually are. So between 0, which is something that I added to this data set, I can see what the difference is between those two, and I can see that that was statistically significantly different. Among the others, though, sites 1, 2, and 3, not much difference. It's really just this site 0 that I added. And then finally, I'll just show you a correlation, so a Pearson correlation, and let's just see if there's a correlation among these 4 different variables that comprised our main outcomes. And it gives me this nice little correlation matrix. I will save this in my output. You can choose if you want the p-value to be single-tailed or two-tailed. You can choose to highlight by p-value, so it'll show you which one of these were statistically significant relationships. But this is really just a standard correlation matrix, and it's what I would expect for this data set. So that is a very quick tour of MAXQDA stats, also bringing in variables, which I consider foundational. And I'd be happy to answer any questions that might have come in. And I'm not seeing any in the chat. I know a few people asked for recordings, which are available. My understanding is that the slides are also available. If you have trouble accessing them, you can always email me, and I'm happy to send them to anyone as well. Put my email address in the chat, if you can see that. Any questions? Okay. Does the ANOVA require specifying the statistical model? No, it is really just a fairly simple one-way ANOVA option. So it's not like you're using a general linear model where you might be having multiple factors involved or something like that. This is really just a single factor with a variety of variables. So that's kind of your only option with this one-way analysis of variance. I hope that addressed your question. Any others? Great. So maybe when I have a minute or so here, I can go ahead and show you a little bit more. I'll show the crosstabs and work in the chi-square test because we really didn't much of a chance to do that. So I'm coming back to compare groups and the crosstab. And then here you can select what you want your rows and columns to be. So let's say I'll keep it as site for rows, and we'll do a categorical outcome variable as columns. And sorry, my Zoom window is right in the way. So here is my crosstabs right here. So really this is just, the intersect, or, the intersection, I guess is one way to look at it, of site by the categorical measure. It doesn't really give you any tests, but if you come to this option, this little icon here, kind of to the left, it's association measures, and I'll click on that. And here, it gives me the actual chi-square, so it does test that for me. And then it gives me a Kramer's v-value as well, and then a contingency coefficient. So it does give me several different associational measures, so I can do that pretty easily. And then the final thing then I'll just show you briefly is the scale analysis. So the Chromebox Alpha. This isn't going to make a ton of sense for what we're doing here, but I'll just use some of these items to show it to you anyway. So I can see if these variables seem to be internally consistent. Again, this makes a lot more sense if you are working with a survey, some kind of scale, but I'll click OK. And then it gives me this reliability analysis. So my overall Chromebox Alpha is 0.847 of how many cases. And then it gives me what the mean scale would be without this item. So if we would remove this item, it gives me how the alpha, so how that coefficient alpha would change without this item. So if I dropped one of these, would it improve reliability? Commonly done with this type of analysis. So it gives me that as well. And, just looking at this, you can see if I deleted any of these, my reliability would actually be worse. My coefficient alpha would go down. But this is really nice to have within the software to run, even sort of a quick test of a survey, any survey data that you have collected. All right. Any other questions? And if not, that's okay. Um, I certainly thank everyone for joining and participating. This is a great conference, and I hope you've benefited from it. And thanks for posting that link as well to additional information on the stats module. But this is something that I use. Again, this is actual data. This is a real data set, something that I've found helpful. Wonderful.